Hello students, in this lecture I am going to talk about the variety of electrodes. Basically there are two types of electrodes, biopotential electrodes and biochemical electrodes. So in biopotential electrodes there are three types. Basically there are three types: micro electrodes, then we have surface electrodes, and then we have needle electrodes. So what are micro electrodes? So these electrodes, in short, if I have to tell you what are micro electrodes, they are used to measure bioelectric potentials near or within a single cell. And skin surface electrodes, they are used to measure ECG, EEG and EMG potentials from the surface of the skin. Whereas needle electrodes, they are used to penetrate the skin to record EEG potentials from a local region of the brain or EMG potentials from a specific group of muscles. So I'll be discussing all these electrodes in detail in my upcoming slides. So let's start with microelectrode. So what are microelectrodes? As I've already told you, these electrodes, they have their tips. These electrodes, they have their tips which is sufficiently small so that it can penetrate a single body cell, a human body cell, so that it can penetrate a single human body cell so as to obtain readings from that from within that cell. And the tip of this electrode, the tip of the microelectrode should be small enough so that it can penetrate easily without damaging the body cell. These microelectrodes are of two types, metal and the micropipette. This is the metal type, this is the micropipette type. So metal electrodes, they are formed by electrolytically etching the tip of a fine tungsten or stainless steel wire to the desired shape. The wire is then coated almost to the tip with an insulating material. Here you can say insulating material is used around this metal. And some electrolytic processing is also performed on the tip so that the impedance can be lowered down. The metal ion interface takes place. This metal ion interface takes place where this metal tip contacts the electrolyte either inside the body cell or outside the body cell. That is, when this electrode is going to penetrate the body cell, only then you'll get to see the metal ion interface. This is the metal, and the ion, that is the electrolyte, that is a cell fluid. Electrolyte is here, is the cell fluid. Next is the micropipette. In micropipette type of microelectrode, there is a glass micropipette. Here you can see there is a glass micropipette with tip drawn out to the desired size, and this micropipette is usually filled with an electrolyte that is compatible with the cellular fluids. It is filled with the electrolyte. Now this type of microelectrode has dual interface, has two interfaces. One interface consists of, this is the metal, one interface consists of metal wire in contact with the electrolyte solution inside the micropipette, while the other is the interface between the electrolyte inside the micropipette and the fluid inside or immediately outside the cell. That is, when this micropipette electrode is going to penetrate the cell, the second interface will be between the electrolyte inside this micropipette and the fluid inside or outside the human body cell. So this microelectrodes, because of their small surface areas, they have impedances well up to into the mega ohms, and for this reason the amplifiers which are used with this micropipettes should have very high impedances so as to avoid loading the circuit and minimize the effect of small changes in interface impedance. So this was about microelectrode. The next is the next electrodes that I am going to discuss. These all are body surface electrodes. So the next few electrodes that I am going to discuss, they all are body surface electrodes. So what are body surface electrodes? As I have told you, these body surface electrodes, they are used for obtaining the bioelectric potentials from the surface of the body. And these electrodes, they are found in many sizes and forms. Although any type of surface electrode can be used to sense ECG, EEG or EMG potentials, the larger electrodes are usually associated with the ECG, since localization of measurement is not important. Now you can see in this slide here, this is also a body surface electrode. Here you can see this is the immersion electrode. In body surface electrode, this is the immersion electrode. So this is the earliest bioelectric potential measurement. In earlier days, the bioelectric potential measurement was done using this immersion electrode, which were simply buckets of saline solution. You can see here, this is the bucket of saline solution. This is the bucket of saline solution. This is the bucket of saline solution. So these were simply buckets of saline solution into which the human has to place his hands and feet, one bucket for each extremity. So this type of electrode present many difficulties, such as restricted position of the subject, that is the human body has to be there in one position for a longer period of time, and the danger of electrolyte spillage. So this was the drawback of this immersion electrode. Now improvement over this immersion electrode were the plate electrodes. As you can see here, these are the metal plate electrodes. So these electrodes were separated from the human body skin by cotton or felt pads soaked in strong saline solution. Again I am repeating here, the electrolytes, they are separate, they are not in direct contact with the human body, they are separated from the human body by cotton or felt pads soaked in strong saline solution. Later a conductive jelly or paste replaced this soaked pads. Later we started using this electrolyte paste instead of this soaked pads, and metal was allowed to contact the skin through this thin coating of the electrolyte jelly. Means the plate electrodes were used over the human body through this electrolyte jelly. So in this then we have this another type of electrode like this suction cup electrode. In this also only the rim, this is the rim, only the rim contacts the skin. Now one of the difficulties of using plate electrodes, as you can see, one of the difficulty of using plate electrode is the possibility of electrode slippage or movement. So it might be possible that the electrode that is placed over the human body through the electrolyte jelly, it might slip or there might be movement, and this also occurs with this suction cup electrode. The same problem occurs with this suction cup electrode after a sufficient amount of time. Now number of attempts were made so that this problem can be overcome, including the use of adhesive backing and a surface resembling a nutmeg grater, which can penetrate the skin to lower the contact impedance and reduce the likelihood of this slippage of electrode. So this all the preceding electrodes they suffer from the common problem, that is they are all sensitive to movement, some to a greater degree and then others. Even the slightest movement changes the thickness of the thin film of electrolyte between metal and skin, and therefore causes changes in the electrode potential and impedance. In many cases the potential changes are so severe that they completely block the bioelectric potentials and the electrodes bioelectric potentials that the electrodes attempt to measure. So the use of adhesive tape and nutmeg grater actually reduce this movement artifact by limiting electrode movement and reducing interface impedance, but neither is satisfactorily insensitive to movement. Then a new type of electrode came, that is the floating electrode, which was introduced in various forms by various manufacturers. So the principle of this electrode is actually what is the advantage of this electrode? It was used to eliminate movement artifact by avoiding any direct contact of the metal with the skin. So there was no direct contact of metal with the skin. The only conductive path between the metal and the skin is the electrolyte jelly, which forms an electrolyte bridge. So as you can see here, this is the floating type skin electrode. This is the diagram of, or you can say the cross section diagram of the floating type skin electrode. Here you can see this portion, this is the metal and this is the electrolyte jelly. Sorry, this one is the electrolyte jelly, which is placed in between metal and the skin surface. This is the skin surface. This is the lead wire, which is going to take the bioelectric potential to the amplifier, etc. Then we have this plastic or rubber support spacer. Okay, so this is the cross section. Now this floating electrodes, they are generally attached to the skin by means of this two-sided adhesive collars or rings. You can see there is two-sided adhesive. You can see this is the two-sided adhesive collar or ring, which actually stick to both the plastic surface of the electrode and as well as stick to the skin. Then various types of disposable electrodes were introduced, which eliminate the requirement for cleaning and care after each use. Just like in the previous electrodes, after each use you need to clean or you need to care the equipment after each use, but here this requirement was eliminated, and it is primarily intended for ECG monitoring. These electrodes can also be used for EEG and EMG as well. So disposable electrodes, they are of the floating type with simple snap connectors. You can see these are the simple snap connectors by which the leads, which are actually reusable, are attached. So these leads, they are reusable, they can be attached through the snaps and they can be removed from this electrode, and this electrode can be disposed. So what is disposable electrode? Their cost is so low that they can be used for one time and they can be thrown away. Okay, the next electrode that I am going to discuss under this body surface electrode is ear clip electrode. So here this was developed for use as a reference electrode for EEG measurements. So this was all about body surface electrode. Next type of electrode that I am going to discuss is needle electrode. So in needle electrodes, what happens? It reduces the impedance and the movement artifacts. Some